Alright, ladies and gentlemen, this is going to be the next video on molecular biology and specifically looking at the types of RNA. Let's get started on this next video. So the function of RNA, well, let's think about blueprints for a second. Let's say I want to go through and I want to make a light bulb. I have a master copy of that blueprint for that light bulb. Well, I want to distribute that to a variety of different people to help me make that light bulb. What I'm going to do first is I'm going to make a copy of that blueprint. I'm going to use the master blueprint here. Some people might spill coffee on it. Maybe I'll get a slice of pizza on my copy, and I want to make sure that I'm not using our master blueprint to go through and perform whatever I'm going through and building. We're doing a light bulb right now. And the way I kind of think about RNA is it's kind of like a disposable section of DNA, right? It's segments that we're pulling out for the DNA, the specific segment that we need. We don't need all the directions for everything. If I'm trying to make proteins in my body for my hair, well, I only need this section of the DNA for my hair. So I don't need the entire section of DNA to do that. And RNA is really disposable, and we're using the genes that are present in our DNA to make small segments of RNA that we then turn into the proteins in specific parts in our body. So there are three types of RNA that we have to know. The first one is going to be rRNA, and this is ribosomal RNA, and it's going to form the important subunits of our ribosome when we talk about the structure and function of this and how the ribosome really works. We also have tRNA, and tRNA transfers amino acids to the ribosome, and it's going to transfer them to that mRNA-coded message. Now, mRNA, well, this is going to be the strand that we get from the DNA, and this is messenger RNA, and this carries the instructions for polypeptide synthesis from the nucleus of the ribosome into the cytoplasm. So let's quickly go through and review proteins. Proteins are made from, well, they're made from amino acids. Amino acids are going to be the monomer. And our amino acids are going to consist of our carboxyl groups and our amino groups. And this is going to create the building blocks for proteins. So here's our amino group that we can remember. Here's our carboxyl group. And each one of these little spheres represents our amino acids. We also have our side chain here, which is going to really differentiate different amino acids from each other. Now, if we're looking at this, each amino acid has this central carbon, and then we have all of our functional groups bonded to the rest of our amino acids here. Now, if we continue upon this, well, we have chains of amino acids that are going to make up our proteins, and they're held together via these peptide bonds. So the peptide bonds are chains of amino acids held together, and then we take these chains and we fold them into the 3D structures, which creates the proteins and animal structure. And we can eat proteins that eventually turn into the proteins in our body, and we're using the ribosomes to go through to break down our amino acids that we get from our food, and then take those amino acids, turn them into peptide chains, and then turn them into the complex folded proteins that we observe in our bodies. So if we're looking at how these molecules work together, we want to figure out how we go through and assemble these proteins in our body. Well, our mRNA is going to meet with our ribosome, and there's going to be all these tRNA strands that are going to float apart. Now, if we look very closely at the top of these tRNAs here, what we're going to see are amino acids. And these are the amino acids that we're going to peptide bond together, and we're going to sequence them based on the sequence of our mRNA. Once we have these peptide bonds sequenced together, we get this long peptide chain that we then fold into our protein. So all three of these pieces form together: the mRNA, the tRNA, and then our rRNA from our ribosome kind of fold together in this mechanism to create these peptide bonds that fold this polypeptide chain here. Now, once we have our peptide chain, we're going to fold up all of our proteins into these complex shapes based on their polar groups and the polar regions. And the environment and the enzymes that they interact with affect the folding. So if we try to piece this all together, this entire process of going from DNA to RNA to then proteins, well, this is called the central dogma of biology. So when we go from DNA to RNA, well, this is what we call transcription. And then when we go from RNA to proteins, and that's that little mechanism that we've gone through and looked at, well, that's called translation. So what we're doing is we're learning the types of RNA, and then we're going to delve into the nitty-gritty of transcription and translation in these complex processes to figure out how we go from DNA to protein. That is the central dogma of biology. So, did you guys learn? Well, did you learn a couple of things? Did you guys learn the types of RNA? Did you guys review proteins? And then did you guys learn the basics of the central dogma? This is going to be the end of the video. I will see you all in class tomorrow.